Hi, Steve. Thanks for taking time out of your schedule to come and have a chat with me today. I thought it would be interesting just to have a little discussion about impurity testing. I know we've got a lot of work that's going on in the lab at the moment with all sorts of impurity studies going on. So I thought maybe you could just tell us a little bit about why impurity testing is so important.
Yeah, thanks for talking to me, Richard. And yeah, impurities are really important to test, and they have very big effects on the safety and the efficacy of your product. And there's really two main concerns surrounding impurities. The first is that impurities can invoke adverse effects in patients, such as immunological responses. And the second is that impurities can have adverse effects on the product itself through modifications to the product, like oxidation or aggregation or truncation, just as a few, for example. And these changes to the product can negatively impact the effectiveness of it and the shelf life. And these adverse effects can be caused in some cases by really low ppm amounts of impurities. And for all of these reasons, it's really important to monitor impurities and use that knowledge to feed back into your production process with the goal of minimizing and reducing impurities wherever possible. And a final point on why it's important is that the regulatory authorities are expecting that impurities have been checked for as a crucial part of the product characterization process.
Sure. Okay. So there's obviously good reason for analysing these things. Obviously, there's a huge range of molecules that people are working on. There's going to be a huge range of processes that are involved in producing these drugs. And that's going to give rise to a huge range of impurities, of course. So maybe you could say something about a little bit about the kind of impurities that we're seeing, that we're working on, and some of the techniques that we use to analyze these.
Yeah, sure. And as you say, there are a wide variety of impurities that need to be checked for, and we test for a lot of them here at BioPharmaSpec. Just a couple examples of some that we're working on right now: inducer IPTG, for example. Kanamycin, which is an antibiotic, and host cell proteins. We actually have a few host cell protein studies at the US lab site in process right now. And as far as methodology goes for how we check for these impurities, we rely on mass spectrometers for the detection. And mass spectrometers provide high sensitivity, allowing for detection of impurities that are often at really low levels. And mass specs provide quantitative data, which not only allows you to assess what's in the sample, but also tell you how much of each impurity is in the sample. Both of those are very important to know when you're assessing impurities.
Sure. Okay. Well, thanks for that, Steve. You touched on one point there that I'd like to sort of follow up on briefly, and that's host cell proteins. Obviously, that's a huge area as far as analysis goes, and mass spectrometry is playing a significant role in analysing these. Could you just say a little bit about how mass spectrometry compares to sort of more classical ELISA methods for analysis of HCPs?
Yeah, sure. I mean, yeah. So as you mentioned, ELISA is a very common and actually considered the gold standard approach to assessing host cell proteins, and it's for good reason. ELISA does a great job of providing low-level detection of host cell proteins and quantitation of host cell proteins. And ELISA is specifically well suited for routine testing, where you need to run the test many times. And if you contrast that with mass spectrometry, mass spec isn't quite as good for these routine tests because the data that's generated with the mass spec analysis are much more complex. You need a lot more expertise to analyze the data, and they're relatively higher cost to run. On the other side, though, mass spectrometry can provide information that ELISA simply can't. Like, for example, mass spec allows for the identification and quantification of specific proteins, whereas you compare that to ELISA can only spit out the total weight of all the proteins present. So it doesn't really give you any information about what those proteins actually are, whereas mass spec can give you that. So I guess in summary, we see mass spec and ELISA both as crucial to the process of analyzing host cell proteins, and we think they're both complementary to each other too. So we suggest doing both tests to really get a good idea of what your impurities are.
Right, sure. Okay, so yeah, so they're very much sort of orthogonal techniques one used to sort of support and expand the other. Okay, that's great. Well, thanks very much, Steve, for taking time to chat to us today. Our contact details are up on the screen right now, so if you have any questions about residual analysis, please do get in touch. And please take a look at the website as well. There's a lot more information on there. And thanks for taking the time to listen in today.